Hi guys, welcome to my YouTube channel Unique Bio Classes based on NCERT syllabus for PU first and second year. If you like my channel, please subscribe, like and share and hit the bell icon so that you can get notifications as soon as videos are getting uploaded. Okay guys, again after a long time I'm here with a new video, so hope you like it. If you like, please like and subscribe. Okay, in my previous video I have discussed about hemophilia. Now I am going to discuss about sickle cell anemia. Okay, the sickle cell anemia you will get in your NCERT textbook in the page number 89. On page number 89 you will get sickle cell anemia. So, sickle cell anemia comes under a Mendelian disorder. Mendelian disorder, we have discussed about hemophilia. Next one is sickle cell anemia. Now, regarding sickle cell anemia, you have on this page and I have prepared a note of it. We shall go with that. What is that note? So, this is it. Okay, this is the sickle cell anemia which I have prepared it here. Now, in the sickle cell anemia, if you find it, okay, let me adjust. Everything is visible. Fine. Okay. Sickle cell anemia, it is an autosome linked recessive trait. Okay, as you know, autosomes are what? These are the other chromosomes. Other chromosomes are what? Other than the sex chromosomes. As we said, every organism is having one sex chromosome, some other chromosomes. Other than the sex chromosomes, what chromosomes are present? For those we call it as autosomes. Sex chromosomes are also called as allosomes. If you talk about human beings do have 23 pairs of chromosomes, right? So in those 23 pairs of chromosomes, one pair of chromosome is a sex chromosomes or allosomes, and rest 22 pairs of chromosomes are called as autosomes. Okay, now in this sickle cell anemia, it is autosome linked, which means it is not related to sex chromosomes, it is related to the autosomes. Other than the sex chromosomes are called as autosomes. Autosome linked recessive trait. Okay, a trait, as we know, it is nothing but a character. Recessive, which is not, which will won't express in presence of dominant. Is it? Recessive will won't express in presence of dominant. Autosome linked recessive trait, it shows that it is not related to male or a female, as we have found in hemophilia, that is getting transferred from parents to offspring, that is mother to son. Is it? From mother to son. But here we are not saying that it will transfer from father to a daughter, or father to a son, or mother to a daughter, or mother to a son. Why we are not saying that? Because it is autosome linked, whereas hemophilia was sex linked disease. This one is autosome linked recessive trait. Okay. Now this sickle cell anemia, it was discovered by Pauling in the year 1949. Okay, many other scientists also have been worked regarding this sickle cell anemia, but most probably Linus Pauling, he has discovered it in the year 1949. Now, it is transmitted from parents to offsprings. Okay, which one? Sickle cell anemia, it is transmitted from parents to offsprings when both partners are carrier for a gene or heterozygous. Okay, so this sickle cell anemia is transferred from parents to offsprings. Okay, which means any of the offsprings, either male or a female. When it is getting transferred, when both the partners are carrier, which means both the parents, right, both the parents, that is father and the mother, should be a carrier for a gene. Okay, which gene? The gene which is here, it is in a recessive condition. Okay, the gene which is responsible for sickle cell anemia, that gene must be in the form of carrier in both the partners. Okay, then only this disease is getting transmitted from parents to offspring. So how it is, I shall show you in detail later. The disease is controlled by single pair of allele. Okay, the disease is controlled by single pair of allele. This allele is present in a gene. As we know, a gene consists of a pair of alleles. That pair of alleles, in that it consists of HBA and HBS. Now what is this HB represent here? HB is nothing but hemoglobin. Okay, HB represent hemoglobin. Here HbA represent, it is a normal hemoglobin. HbS represents abnormal hemoglobin. Okay, it is not normal. Now the disease is this sickle cell anemia is controlled by a single pair of allele or a single gene with a HbA and HbS. Fine. So if you talk about this, now this is one of the chromosome. If I'm showing it here like this is one of the chromosome. This chromosome consists of one of the gene. That gene consists of pair of alleles, and that pair of alleles, it can have HB. It is having HB. Now this gene is responsible for production of hemoglobin in that human body. Okay, whoever's body it is, in that it is this gene, it is alleles, or this gene is present used to produce hemoglobin. Okay, now this a pair of alleles HBA, 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 if it is in this condition, then that is normal. Okay, it is normal. If these are pair of alleles, right? Even alleles are present in a gene, both the allele, if they are normal, then the individual will be normal. How normal? Which means normal hemoglobin will be produced in that individual. If this hemoglobin alleles are in this condition, HBS, HBS. Now S represents sickle cell anemia here, which means it is abnormal. Now this one is affected individual. Okay, this one is affected. Fine. If we are having this condition, HBS and HBA, if we are having this, one allele is sickle cell, another allele is normal. Okay, one is normal, another one is having the sickle cell allele. So in this one is heterozygous, right? Heterozygous means what? Two different types. Different. Homozygous means same, heterozygous means different. So two different types. These are homozygous. This one because both the alleles are same. This one is homozygous because both the alleles are same. This, if this is present, is normal. If this homozygous condition is present, then it is affected. Affected means that person is having sickle cell anemia, or he is deceased, and he or she is deceased. And if they are having different types, HBS and HBA, then it is heterozygous, or we call it as carrier. Okay, because this individual is carrying a sickle cell anemia. This individual is not called deceased or affected. Why? Because here HBA is present. This HBA, it gives that person to produce normal hemoglobin. This will be suppressed. HBS will be suppressed by this HBA, because this HBS is recessive and HBA is dominant. Now this dominant one will suppress the HBS, so that this affected one will be, okay, the affected one will not function, whereas normal one will function to produce normal hemoglobin. Fine. So this will be produced here in this hemoglobin in this sickle cell anemia. Okay, we were here. The disease is controlled by single pair of allele HBA and HBS. Okay, now for that I have given here, if you find further, here I have given one column of phenotype, another column of genotype. Deceased, its genotype will be what? HBS, HBS, as just now I have shown there, and the carrier, it will be having HBA and HBS. Okay, this one is carrier, one is normal, one is sickle cell, and the one is the normal individual. If a normal individual, what does they have? HBA, HBA. This is how they are going to have the different genotypes of different phenotype: deceased, carrier, and normal. Now you got to know what is carrier, what is deceased, and what is normal. We shall come back to the second statement which we have discussed here. Sickle cell anemia, it is transmitted from parents to offsprings. Okay, parents to offsprings, when both the partners are carrier for a gene, or heterozygous, when both the partners should be a carrier. It has mentioned as carrier. Okay, so carrier is what? As just now we have seen, carrier, its genotype will be this. Both the partners should have this. We shall try to cross that one here. Okay, we shall give one side female and other side as male, both the partners. Okay, now both the partners should be carrier. We have given HBA, HBS, and here also HBA, HBS. Fine. Now they will produce a gamete. It will produce HBA, HBS. These are the two different types of gametes. Similarly, same gametes will be produced. Same type of gametes. Male will produce sperms, female will produce eggs. Right? That story is completely different. Here, if this gamete is fusing with this one, what you will get? HBA, HBA. Right? If the same one is fusing with the next one, what you will get? HBA, HBS. Then you will find this HBA and HBS. After this you will find HBS and HBS. These are the 4 different types you will get. Okay, now we are not saying which one is male, which one is female, how many male offsprings, how many female offsprings. We are not saying. Why? Because it is not related to sex linked, it is related to autosomes. We know that 4 offsprings can be formed, different types, and what all are the chances, we can see that. Okay, HBA, we said it is normal, so this individual is normal. HBS is what? Carrier. HBS is what? Carrier. Right? HBS is what? It is affected. Fine. So what that statement has said? When we are going to get affected? When both the partners are carrier. When both the partners are heterozygous. In this condition only we can get it is affected. If we are getting one parent is carrier, another parent is affected, what are the chances? Whether we'll get children's affected or no? Yes, definitely we'll get children's affected. Okay, so this one you will get, these are the 2 different types of gametes, right? HBS, you will find these. Okay, now if you cross this one, you will get HBA, HBS. Okay, then you will get HBA, HBS. Then here HBS, HBS. After this you will get HBS, then HBS. Fine, you will get this. Now this one, what will say? This one is carrier, this one is carrier, this one is affected, and the next one is also affected. Okay, what we said, when both the partners are heterozygous, then we'll get affected individual, then an offspring can get affected by it. If one partner is carrier, another partner is affected, then also we can get affected individual. Okay, at least both the parents should be carrier, both the parents should be heterozygous, at least they must be carrier. If they are affected, then definitely will get, if one is affected and one is normal, if one is normal, one parent is normal and another parent is affected, what do you say? What kind of children will get? Whether will get affected or no? Okay, I want this answer. Okay, whether you will get affected children or no, I want this answer. And if one is carrier and other one is normal, HBA, HBA and HBS, HBA. Okay, what children will get? What children means how many carrier, how many affected and how many normal. I want this. Please comment it to the comment box so that I'll come to know whether we have understood this concept or no. Okay, if a normal individual is getting married with a affected individual, whether will get affected children, I want this answer. Another one condition is that whether if a normal individual is getting married with a carrier individual, then how many carriers, how many affected, how many normal. I want this answer. Please comment it in the comment box. Okay, so the condition is what it has been said, it is transmitted from parents to offsprings when both partners are carrier, when both partners should be a carrier for a gene or they must be heterozygous, right? So hope you understood this sentence. And we have given about this, the disease is controlled by a single pair of allele, HB and HBS, and we have discussed about this also, phenotype and genotype of it. Okay, now the defect is caused by the substitution of glutamic acid. Why does this sickle cell anemia will occur? Okay, the question is that the sickle cell anemia is going to occur because of substitution of glutamic acid by valine. Now the question is, what is this glutamic acid and what is this valine? If you have gone through your first year, in that you had about this one in biomolecules, that glutamic acid and valine are nothing but amino acids. These amino acids are used to form proteins. Okay, now hemoglobin is what? Hemoglobin is nothing but a protein. Now that protein is actually made up of glutamic acid and valine. Not only this, it is produced by many different types of amino acids. Okay, now this glutamic acid by valine at the 6th position of beta globulin chain of hemoglobin molecule. Now hemoglobin molecule, it do have presence of alpha chain and a beta chain. Now in the beta chain, the chain, if you find, I will show you that one in the textbook. In the textbook you will find it very clearly. Okay, it has been shown here. Now this one you have, okay, this one, it has been mentioned it as HBA peptide. Fine, this one is a HBA peptide. HBA peptide means hemoglobin peptide, a normal hemoglobin. Okay, peptide is what? It is nothing but protein. Fine. When sequence of amino acids are present, we call it as a protein. Okay, now this is its structure. Simplest way they have shown the structure. Actually, it is in very complex form. Simplest way, easiest to understand with you people. This is normal HBA gene. Here you can find GAG and CTC. Now what are all this? You will understand in the next chapter. If you know about molecular basis of inheritance, then it was very easy to understand this. But for time being, I shall just go through overview of it so that you can understand now. In detail, if you go through in the next chapter when I am dealing with those chapters, those topics, then it is very easy to understand. Now this is the DNA. Okay, this is the DNA. As you know, DNA consists of nucleotides. So GAG and CTC, it's complementary, it will be present. So this is the DNA. From DNA, mRNA is produced. Okay, this is the mRNA. mRNA is utilized to form a protein. Okay, so transcription and a translation will take place here. I'm just giving an overview. I think you may not be easily understood. If you have gone through molecular basis of inheritance, then definitely it will be very easy for you all to understand. Okay, now this is a HBA peptide, hemoglobin A, hemoglobin chain of a beta globulin chain, which it do have presence of first amino acid as valine, second one as histidine, third one leucine, fourth one threonine, fifth one proline, sixth glutamic acid, seventh glutamic acid. Okay, this just 7 amino acids they have given. Why they have given? Because 6th position is altered, it has been changed to show that it has been shown here as glutamic acid. Fine, now it has been given in the short form of it. Okay, amino acids are represented by in this short form, or it is also represented by alphabets also, like A, B, C, D, like that also it has been and it has been mentioned. But for you all, it has been given simplest way in a short form and a name of the amino acids. In higher classes, you will come to know what does A represents which amino acids, B represents which amino acids. You will study in your higher classes. Okay, now this you come to know, this is the normal hemoglobin beta globulin chain. Fine. Now here you will find there is presence of HBS peptide, which means it is sickle cell anemia. Okay, it's hemoglobin, it's peptide. You can find same sickle cell HBS gene, GTG, CAC. Here what it is? GAG. In place of GAG, here it has been replaced as GTG. In place of A, it is T. So in place of T, here it is A. Okay, because it has been changed here, then you can find mRNA GUG has been formed. Here it is GAG, here it is GUG. Because of that, the protein which is produced here, the amino acid which has been produced here, first one is valine, second histidine, third leucine, fourth threonine, fifth proline, sixth valine, seventh glutamic acid. Can you find a change in this sequence from this normal one? Definitely you will find valine, histidine, leucine, threonine, proline, glutamic acid. Can you find at 6th position? Here it is glutamic acid, here it is valine. 7th one is glutamic acid, 7th one is same as it is. So what does it says? At 6th position of the glutamic beta globulin chain, glutamic acid is replaced by valine in sickle cell anemia. Understood? So I shall show you my note of it, what we have said here. The defect is caused by the substitution of, one second, let me adjust it to camera. Okay, The defect is caused by the substitution of glutamic acid by valine at the 6th position of beta globulin chain of hemoglobin molecule. Hope you understood this. Okay. Why does it take place? It results due to single base substitution. Okay. Why it occurs? Because single base substitution. Substitution means, as you know, replace. Is it? Single base substitution at the 6th codon of beta globulin chain from GAG to GUG. We have seen this. Right. For this we call it as codon. In the next chapter we are having that. From GAG it has been replaced to GUG. Because of this, we have got change here, the change of the amino acid. If same GAG was present here, we would have got the same amino acid. Just because in place of A, it has been placed as U, we have got different amino acid. That sentence is here. It results due to single base substitution at the 6th position of beta globulin chain from GAG to GUG. Okay. This mutant hemoglobin undergoes polymerization. Now this hemoglobin has been changed. Normal hemoglobin is different. Now this hemoglobin is different. This mutant hemoglobin undergoes polymerization under low oxygen tension. Okay, polymerization, it will change its shape under low oxygen tension, because hemoglobin function is to carry oxygen. Because it is not having a proper structure, hemoglobin does not have a proper structure, because so it could not carry the enough oxygen, 4 molecules of oxygen. So it will change its structure, causing change in the shape of RBC. Okay, RBC shape is what? Biconcave shape, but here it is getting converted to sickle shape. Okay, here because of low oxygen tension causing change in the shape of RBC from biconcave disc to elongated sickle-like structure. Okay, from elongated sickle-like structure. Now what is the sickle-like structure? Sickle, I shall show you in the picture. In the next you will find what is meant by sickle. That sickle actually is used by the farmers, even by us, to cut something in the grass or something. That sickle you will find in the next picture. And because this RBC has changed its shape to that structure, so it is called as sickle cell anemia, because cell has changed its shape. So it is called by that name, it has called as sickle cell anemia. Okay. This is sickle because the RBC have changed their shape from biconcave disc-like to this sickle-like shape. So it is called as sickle cell anemia. See, this you can find. This is the sickle cell RBC. Okay, because it has been changed its shape and hemoglobin could not transfer enough oxygen, so it is called as sickle cell anemia. What do you mean by anemia? Anemia is nothing but where less amount of hemoglobin is present in the body. When less amount of hemoglobin is present in the body, we call it as anemia. And because of change in the structure of hemoglobin, definitely there will be a less amount of hemoglobin, which is not enough to transfer enough oxygen in our body. Okay, so this is about sickle cell anemia, very important and very easiest. If you like this one, then you can copy the note, and the note which I have prepared, it is directly from your NCERT textbook, nothing new. Just it might help you all for understanding, and hope you like it. And I shall try to add some more pictures in this video, so that it is very easy for you all to understand. And how does these sickle cell anemia will block the capillaries, that also I will show in my next picture. You will find that too. Now in this picture you will find the normal red blood cells and sickle red blood cells. The sickle red blood cells, how they are blocking the flow of blood in the capillaries, that is also clearly visible. You can just compare and you can understand it. Now they might have started their preparation, so share this with those students also, it might help them. In future I am trying to upload videos. Hopefully in my next video I may deal with phenylketonuria. Okay, stay safe, stay healthy.